started.
Good evening, everyone. Welcome to the final webinar in the webinar series organized by the Staff Development Center, Nitte deemed to be university, in association with Nitte Center for Integrative Medicine and Research to celebrate the National Nutrition Week. I'm Sadhana Deshmukh, Director, Staff Development Center. Welcome you all. We also welcome Dr. Kumar Kothapalli, who is going to be the speaker for today, and he is going to talk on genetic aspects of omega fatty acids. I'm very sure all of you are interested, eagerly waiting to listen to his talk. I also welcome Dr. Arun Tejas, who is in charge of Nitte Center for Integrative Medicine and Research, and also welcome all the delegates and students who have joined this webinar to get more information on this subject. I request Dr. Arun Tejas to introduce today's speaker, Dr. Kumar. Over to you, Dr. Arun.
Good evening, everyone. It's a great privilege for me to introduce Kumar sir. He has done a good work on omega-3 and we are eagerly waiting to listen from him. And he has done his graduation and post-graduation in India and his PhD in medical genetics from Osmania University. Then he has moved to US to do his postdoctoral fellowship in genetics and genomics from Cornell University. At present, he is working as an associate professor of pediatrics, Dell Medical School at University of Texas, Austin, US. Last 1.5 years, he was working in the field of omega-3 fatty acid and omega-6 fatty acid related research. And reason his work has been greatly gone viral, like about the balance of omega-3 fatty acid and omega-6 fatty acid. We know that omega-3 fatty acid and omega-6 is an essential fatty acid that has to be get into our system through our food, and he will now give a brief idea about how genetic related to this fatty acid. And thank you very much, sir, for joining with us and enlightening us about this subject. I welcome you, and over to you, sir.
Thank you so much, Dr. Arun, for a great introduction. Thanks so much, Sadhana Deshmukh ji, for inviting me to give this talk. So, can I share my slides, please? So today I would like to talk about the genetic aspects of omega-3 fatty acids, especially omega fatty acids 6 and 3, and their status in humans based on the genetics. So before starting omega-6 and omega-3 fatty acids, I would like to give a brief introduction about what are these fatty acids. Polyunsaturated fatty acids, any fatty acid which has a chain length of 16 carbons or above with at least 2 double bonds. These are called PUFAs. Saturated fatty acids, they don't have any double bonds. Whereas monounsaturated fatty acids, they have single double bond. So why these polyunsaturated fatty acids are important? Because vertebrates, even humans, we are not able to biosynthesize them de novo. So where do we get these fatty acids from? These fatty acids we get from our diet. That's the reason why the linolenic acid and alpha-linolenic acid, they are considered to be essential fatty acids. They are 18,3N3 and 18,2N6. So once we have this in our diet, we have the genes to metabolize them to make physiologically important longer chain polyunsaturated fatty acids. So once we have essential fatty acids in our systems, they can be elongated or desaturated Desaturation means the enzymes which add double bonds into the fatty acid chain. Elongases, they elongate the carbon fatty acids. So once we have essential fatty acids in our diet, we can make longer chain PUFAs, like arachidonic acid, which is omega-6 fatty acid. EPA and DHA are another most physiologically important omega-3 fatty acids. So how does these fatty acids get their name? Because if you see the methyl or omega end here, the first double bond is at the 6th position. So they are omega-6 fatty acids. When we see the omega-3 here, we have the double bond at position 3. Omega-3 fatty acids, they are required for optimal health, growth, vision, and brain development. If we see here, during the brain growth spurt, especially during the last trimester of pregnancy and up to 18 months or 2 years of age, the DHA gets into the brain rapidly. Now the new data shows that the accumulation of DHA can go up to 18 years and above. These are major structural fatty acids in the cell membranes. They are precursors for cell signaling molecules, especially eicosanoids and docosanoids. Eicosanoids are basically synthesized from arachidonic acid and EPA, whereas docosanoids are synthesized from DHA. And these fatty acids, they are natural ligands for several nuclear receptors. And once we ingest in the diet, they regulate the genes within minutes. So if we see the omega-6 and omega-3 biosynthesis pathway, once we have the essential fatty acids, 18,2-N6 and 18,3-N3, we have enzymes to metabolize them to DHA. So both omega-6 and omega-3, they compete for the same enzymes. So once, when we evolved as humans, we evolved with 1:1 ratio of omega-6 and omega-3. But due to industrialization, we are having a lot of omega-6 in our diet. That means the balance between omega-6 and omega-3 is getting disturbed. Especially if we see in Western countries and in India, the ratio has become omega-6 to omega-3 is like 10 or 20:1. So once we have 18,2 and 18,3, just a second, please. I am getting my pointer. So once we have 18,2 and 18,3 in our diet, we have a gene called FADS2. This introduces double bond at delta 6 position, delta 8 position, and at delta 4 position. So FADS2 is a multifunctional enzyme which has a substrate specificity for at least 16 substrates, not only PUFA or long-chain polyunsaturated fatty acids, but it has a substrate specificity for saturates, monounsaturates, and even branch-chain fatty acids. Till very recently, till 2009, what we have seen is once 18,2 is here, it can elongate in an alternate pathway, and 20 carbon 2 N6 was considered to be a dead end product. But in 2009, we found that FADS2, it can desaturate at 8 position, and it can enter into the regular metabolic pathway by biosynthesizing DGLA. DGLA is a 20 carbon 3 N6 fatty acid, and it is considered to be an anti-inflammatory. It can produce anti-inflammatory carcinoids. Whereas arachidonic acid, it has, it is vastly known as pro-inflammatory, and EPA is known as anti-inflammatory. So the pathway on the both sides, omega-6 side, arachidonic acid is largely biosynthesized as pro-inflammatory, pro-coagulation, pro-anti-arrhythmic, and anti-vasodilation. Whereas EPA does anti-inflammation, anticoagulation, anti-arrhythmic, pro-vasodilation, and E-series resolvents. Whereas DHA does docosanoids and D-series resolvents, protectants, and maricins. And once in this classical pathway, in 2006, there has been some genetic polymorphisms has been discovered in FADS2, FADS1 here, and even FADS3 genes. Initially, we thought like delta 6 desaturation by FADS2 is considered to be the first rate limiting step in the biosynthesis of long chain PUFAs. But due to the genetic polymorphisms, each step, because of the genetics, each step in the pathway is considered now to be rate limiting. So based on the polymorphisms here, it can be a rate limiting step from 18.2 to 18.3, 18.3 to 18.4, whereas we have seen genetic polymorphisms even in elongation genes. They can also limit the biosynthesis. And even here, if we see FADS1, there are genetic polymorphisms, and certain individuals who have certain genetic polymorphisms, they can do faster desaturation from 20:3 to 20:4. And we have seen this LCPUFA pathway, which has been worked on in rodents. It shows like one round of elongation from here, then FADS2 desaturation doing delta 6 to make 24 carbon 5 and 6 fatty acids, and then beta oxidation to make 22 5 and 6. Similarly, in the omega-3 side, we have seen like beta oxidation to make DHA. But in 2015, we published a study showing by using a D5 ALI to the pathway does follow delta 4 desaturation. So it does not need a beta oxidation step, but in human cells, we have shown this can do direct delta 4 desaturation to make DHA. So the take-home message from this slide is that DGLA, arachidonic acid, EPA, eicosapentaenoic acid, and DHA, docosahexaenoic acid. These are the most physiologically important fatty acids in our body. They are absolutely needed. We should balance, pro-inflammation and anti-inflammation, to have a good homeostasis. As I have spoken, like fatty acid desaturators, they introduce double bonds within the carbon atoms. So in humans, we have FADS1, FADS2, and FADS3. These are the 3 desaturators. They localize to chromosome 11 at long arm 11q12 to 13.1 region. And these are clustered in 100 kb region within the 11q region. We know the physiological functions of FADS1 and FADS2. FADS3, we have worked out its biosynthesis in the PUFA pathway. But recently we have shown that it's not involved in the PUFA pathway, but it's related to sphingolipid biosynthesis pathway. All 3 genes are evolved by gene duplication events. They have 12 exons and 11 introns. They are transmembrane proteins with well-conserved cytochrome B5 domain and 3 histidine boxes. And FADS genes are extensively and the splice variants are evolutionarily conserved. So several studies involving the SNP studies, largely means single nucleotide polymorphism studies, they have shown that FADS genes are involved with PUFA levels or long chain polyunsaturated fatty acids, or we call it a HUFA also, means highly unsaturated fatty acids. They're associated with HUFA levels and disease phenotypes. If you see like FADS1 and FADS2, FADS2 does even number desaturation like 4, 6, and 8, whereas FADS1 does odd number desaturation like delta 5 and delta 7. In the field, most of the people know like FADS1 does only delta 5, but recently we have shown that it has a delta 7 desaturation capacity. So polymorphisms are associated with LCPUFA levels, especially arachidonic acid. They are associated with certain disease phenotypes, especially cardiovascular disorders and levels of blood lipids, atopic disease, insulin resistance, intelligence in children, attention hyperactive disorder, perinatal depression, and gestational duration. So if we see the common genetic variations, this is based on 1,000 genomes with 88 million genetic variants. It shows like single nucleotide polymorphisms are the large number of genetic variations that have been seen in human. there are certain differences between a polymorphism and a mutation. If in polymorphism, it is present in greater than 1% of the population, but mutations generally are less than 1% in the population. If we see here, C is the major allele and T is the minor allele here. And in disease phenotypes, the similar trend can be seen like C and T. And apart from the single nucleotide polymorphisms, there are several other well-known polymorphisms in humans, which are called insertion deletions. And this is the second largest variations that are seen in human. And large part of my talk will be on a 22 base pair insertion deletion variation, which we found a few years back, which is related with the PUFA biosynthetic pathway. And there are large other variations, which are large structural variations, which are greater than 10,000 base pairs or larger. If we see this classical example of sickle cell anemia, one single base change from here causes a sickle cell phenotype. One single nucleotide out of 3 billion base pairs. So here we are not talking about any disease phenotype, but here we are talking about a polymorphic variations, which can cause how PUFA can be metabolized, either based on the insertion deletion polymorphism, what I will be showing in few slides down now. It shows like, we can have, based on the Indel genotypes, we can have 3 metabolic phenotypes: fast metabolizers, intermediate, and slow metabolizers. So here, an insertion deletion means if we see here a maternal allele and a paternal allele, we can see like this is an example with 4 base pair Indel. Both alleles have 2 4 base pair insertion deletions here. But whereas this is called insertion genotype, whereas here we are missing 4 nucleotides in one of the allele, paternal allele, this has an insertion and a deletion genotype. And in this case, we have 4 base pair deletion in both the alleles, which is called deletion by deletion genotype. So insertion, deletion, deletion. Insertion, and deletion are homozygous, and this is a heterozygous phenotype. So if we see FADS1 and FADS2, the recent gene cards database shows that this FADS1 has around 6,583 single nucleotide polymorphisms, including Indels, whereas FADS2 has 19,669 SNPs and Indels. In 2006, there has been first paper showing like, the genetic polymorphisms associated with this FADS1 and FADS2 gene. Especially when I showed the pathway, we have the classical pathway here, 18-2, 18-3, 23, and then alternate pathway, which is followed by elongation and then delta-8 desaturation. First paper shown by Schaefer et al., it shows that individuals who carry the minor alleles of 11 FADS SNP genotype, they have, increased serum fatty acid levels of 18-2, 20:2, 20:3, whereas they have decreased levels of 18-3. That shows that these individuals, these minor allele carriers, are using alternate pathway than the classical pathway. Especially why this is important is because this is present in 26% of the participants. And till this phase, we see increased levels of fatty acids in these minor carriers. But the next step, which is the delta-5 desaturation, we see like arachidonic acid levels are lower in these individuals. That means this step, the delta-5 desaturation step, these individuals are, not making much arachidonic acid. So in FADS1 gene, there are several polymorphisms that has been found, but one thing which interests a lot of people is an SNP, which is not found in FADS1 gene, but it's upstream in a gene called myelin gene regulatory factor. And this has been shown to be associated with arachidonic acid levels and DNA methylation. Especially the GG genotype of this polymorphism is present in high frequency in African American population. This genotype is associated with enhanced conversion. That means these individuals, they have with the GG genotype, they have higher levels of arachidonic acid. We have done large work on FADS2, especially the intronic region, Intron region 1 of FADS2. This region has been shown to be associated with PUFA levels. Not only with the longer chain, but this has been shown, especially the Indel what we have shown. This Indel, it does bidirectional regulation. That means it is causing FADS2 gene to go up as well as FADS1 to go up. That is very important because it's present in the intronic regions. That means regulatory region. This is a regulatory region, and regulatory region is controlling both the expression of FADS2 and FADS1. I will show the data in the later part of my talk. So in a few years back, like a decade back, we have done a study using Japanese population, samples from HapMap project, where we found like a 10 SNP haplotype present in, especially if we see the block 1 is present in FADS1 and block 2 is in the FADS2 gene, which is this 10 SNP haplotype, which is present in block 2, is associated with the FADS1 level, not the FADS2 level. It's associated with the FADS1 level. That is which is interesting. So this 10 SNP haplotype present in FADS2 is controlling FADS1 expression. So individuals who are homozygous for the minor alleles in this Japanese HapMap, they showed lower FADS1 expression. And then we were curious to see if there are any conserved regions in this intron 1 of FADS2, and we found like this region has the predictive binding sites for sterol regulatory element binding sites and PPARG, peroxisomal response gamma elements. Then we took the lymphoblasts from this homozygous and homozygous individuals who are major and minor, and then we did the gene expression studies. If we see here at the basal levels, the major haplotype and minor haplotype, minor showed lower expression of FADS1, but when we treated them with simvastatin, which is a cholesterol lowering drug, and then LXR, liver X receptor agonist, and rosiglitazone, this rosiglitazone, it did not do anything. But when we treated with simvastatin and LXR receptor agonists, if we see the basal expression of this minor allele, it just matches with the major. So it upregulated the expression of the FADS1 in the minor allele homozygotes. Similarly, if we see the FADS2 expression, there is no difference between a major and minor alleles in FADS2 expression. But when we treated with the simvastatin and LXR agonists, you see the expression in minor alleles, it increased. There is no difference in rosiglitazone. This is PPARG agonist. So that means the cholesterol and the LXR, they are doing something to the expression levels of these minor homozygotes. Then we were curious, we have seen SREP binding sites in the Intron 1. So we were curious to fine map the region. That means to narrow down the region to see if there are any differences in the genetics aspect of these individuals. To the surprise, we have seen a 10 base pair highly conserved sterol response element in the Intron 1 of this FADS2 gene. So if we see this sterol response element, this is, it binds in the DNA. And this is the same, 10 base pair consensus sequence that has been shown by Goldstein and Brown, who discovered cholesterol biosynthesis pathway. Exactly similar SRE is found in the FADS2 Intron 1. All minor homozygotes they have 22 base pair deletion in both the alleles, whereas all the major haplotype homozygotes, they carried a 22 base pair insertion. If you see here in this chromatogram, 22 base pairs is missing, whereas in these individuals, 22 base pair is present in both the alleles. So our hypothesis is individuals carrying the DD genotype have lower metabolic capacity to produce LCPUFA from precursors than the II individuals. Then we, to prove our hypothesis, we need samples from a vegetarian population, predominantly India, and a US population. So what we did was we had a collaboration in Pune University, where we had 234 samples from India and 311 samples from US. This was the initial study. What we have observed is in India, II genotype, which is considered to be a fast metabolizer genotype, is present in 68%, whereas DD genotype is present only in 3%, and the ID in approximately 30%. Whereas if we see in US, the II is present in 18% only, ID in 39%, and DD is in 43%. There is a vast difference between the genotypes in both the Western population in US, these are European mostly, and then Indian population. So due to our vegetarian dietary practices for several generations, II genotype is highly conserved in our Indian population. Then, we started to probe 1,000 genomes because, 234 samples are not that much. But when we saw the different populations from throughout India, Gujaratis, Punjabis, Bengali, and then Sri Lankan Tamil population, Tamil speaking, and then Telugu population, the similar trend was found. In South Asians, approximately 70% are II genotype, whereas in the Europeans, it's only 17%. This we published in 2016, in Molecular Biology and Evolution. And this got a very huge press coverage, which I will be showing later. Then we went on to check the fatty acid levels in these, in US cohort, especially from the Kansas. If you see here, this is the DD genotype and this is the II genotype. The precursor for longer chain PUFA is lower, whereas the product is higher in II genotype. That means it is rapidly desaturated to arachidonic acid. So this we have seen, like, the arachidonic acid levels are 31% higher in II genotype and 13% higher in ID genotype. That is, both homozygous and heterozygotes, they are having higher arachidonic acid levels when compared to the DD genotype. So if we recall how the SNP haplotype difference was, it was 29%, but by using the Indel, now we have shown like the genetic variation increased up to 31%. Then we went on to do some, evolutionary analysis, and we have shown, like, especially in South Asian population, South Asians, this II genotype is adaptive. This insertion allele is adaptive in South Asian populations. Once we have done the study from India and US, we went on to do a larger study from Chinese population using 1,504 Chinese data. This has shown, like, this DD genotype is present in up to in Chinese population, whereas II is in 45%. Then if we see here, in South Asians, the II is very high and the DD is very low. Fatty acid analysis on these Chinese population data. Then here, after doing our analysis, with the larger population sample size, we have seen, like, in II genotype, the arachidonic acid is 57% higher when compared to DD genotype. Similarly, if we do the the ratio of arachidonic acid, which is an omega-6 fatty acid, and EPA and DHA, which is omega-3, we have seen like this ratio, arachidonic acid to EPA, is 32% higher in II individuals. So this data is very consistent with the smaller sample size of 200 samples fatty acid data from Kansas City in 2016, and our follow-up study with larger sample size. Pretty much both shows the similar trends. What is the take-home message is, are we what we eat? Yes, because we are what our ancestors have ate, right? If we see here the DD genotype, we have a less inflammation capacity here. But in II individuals, if we are not balancing omega-3 to omega-6, we might see more pro-inflammation. That is, these are the individuals who are more prone to getting a inflammatory thing, much more when compared to the DD individuals. So our paper, our initial paper, which was large number of media covered our paper on the genetic polymorphism, especially from the Indian data. And the Indian press was very huge on this. And later on, we have to give a clarification that a vegetarian diet does not increase cancers, which the press has misunderstood previously. What we told is the balance between omega-6 and omega-3 should be improved in Indian population, because now the balance is shifting towards 20:1. That means the ratio of omega-6 and 1 is omega-3. So what we told in that paper is like to balance between omega-6 and omega-3, especially for individuals with II genotype. So if we see the intronic indels, these indels not only present in their previously seen associated with some of the disease phenotypes, especially angiotensin converting enzyme. There is a 287 base pair indel, which has been shown to be associated with serum ACE levels. Similarly, a 25 base pair indel in myocardial binding protein C is seen to be associated with cardiomyopathy, especially in South Asians. Now, we do large trials, randomized control trials based on, not taking account of genetics. But recent data is showing like, if we randomize based on the genotypes. So if we randomize the data based on II, ID, and DD, we might get a different response while considering the population to be homogeneous. We need to be genetically, taking account. So from these, my data, what is like a much higher II genotype frequency is found in Indians, followed by Chinese, and compared to US. US, we are seeing only 18. Evolutionary competition tests point to positive selection surrounding this genotype in South Asians. When we compare the omega-3 to omega minus linoleic acid, which is the precursor for arachidonic acid, it was shown like 31% is higher in II individuals and ID individuals compared to DD. We have seen a large follow-up genetic study from China, which shows like II individuals compared to DD. Plasma concentrations of AA and the ratio of AA to omega-3, EPA, and DHA is 57% and 32% is higher, respectively, for these II genotypes. Indels are as important as SNPs in genetic studies. Randomized control trials should take into account genetic variation. So now we are all in a pandemic phase. There is data now coming up showing importance of omega-3 fatty acid, especially resolution of inflammation in COVID-19. We have also published one paper review article recently. In coming 4 slides, I will talk about like how these omega-3 fatty acids can help in reducing the inflammation in COVID disease. This is a paper from HapMap. It shows like, we are all now talking about cytokine storm, or because of the elevation of IL-6, IL-1 beta, and TNF alpha. But before cytokine storm starts, icosanoid storm starts. That means you have arachidonic acid is the precursor for most of the prostaglandins, right? Including thromboxanes and leukotrienes. Thromboxane is involved in thrombosis, which is one of the most frequently seen cause of COVID-19, inflammation and thrombosis. So now with balancing the omega-3 to omega-6, we will have a system where we can achieve resolution phase. That means reducing the inflammation. So now, before talking cytokine storm, we should talk about icosanoid storm. That means prostaglandins, leukotrienes, and thromboxanes, which leads to cytokine storm. Then here, if we see, this is it is shown like increased pro-inflammatory lipid mediators derived from omega-6 arachidonic acid may cause an icosanoid storm, which contributes to uncontrolled systemic inflammation. So early phase, we are seeing inflammation. During the pulmonary phase, we see arachidonic acid going high, leukotrienes and prostanoids. And in stage 3, it's hyperinflammation. So we need to curtail this inflammation by EPA and DHA. We can do that. Similarly, we have published a paper in PUFA, which is a prostaglandin, leukotrienes, and essential fatty acids, where we have seen the difference between 84% ratios, high in these genotypes, in our genotypes, like II individuals. And there is a study from Will Harris group, which showed like, the omega-3 index of 8 and above is considered to be healthy index, 4 and below is considered to be unhealthy index. The people who had lower omega-3 index, they died of COVID-19, whereas who had higher. This is a very small study with 100 people only, but we need a larger study. But this small study has shown like, the benefits of omega-3 index, having higher omega-3 index, which is more beneficial in COVID disease. So I would like to conclude my talk by giving acknowledgements to my long-term collaborator and colleague, whom I have learned a lot of fact. He's Professor Tom Brenna. He is a former president of ISFOL, and he is in the dietary guidelines he was in the dietary guidelines between 2015 to 2020 in US. He is my long-term collaborator. We are working on this omega-3 fatty acids pathway for over close to 2 decades now, 20 years now, close precisely from 2004. And Dr. Park, now he's an associate professor in Korea. He has done a lot of work on omega fatty acid pathway. And now another Park called HiQ Park, he is doing, he's carrying out the legacy and he's doing the pathway. They are our colleagues now. He's now assistant professor in Dell Medical School. And I would like to thank all the authors in these 3 papers who have contributed much to this work. And thank you so much. And I would take any questions from the audience. Thank you. We cannot hear Dr. Arun. You are muted.
Hello, sir. Thank you for that wonderful presentation. I have a question. So you said something related to GG, which is high in Americans and Africans, which means they have much inflammatory problems.
Yes, the GG genotype of yes.
Yes, and you also mentioned about Indians II, which is high, which means we have better conversion ratio and we have better balance or bad balance.
Oh, it's a very good question, actually. So, God has given us a very good gift, actually, II genotype, because If we do a balancing diet, that means if the omega-6 and omega-3 are balanced, because we are not having the longer chain in our diets, right? Because we are not eating fish regularly.
Yes.
So fish has already DHA and EPA, it's already formed.
Yes.
But to make a EPA and DHA, we need a metabolic, very strong metabolic pathway. So II metabolic, the genotype has a good metabolic conversion if we have, the precursors, especially the essential fatty acid precursor, 18.2 and 18.3. If they are in balance, II genotype is the best. But due to higher amounts of omega-6 in our diet, because the enzymes compete, the same enzymes compete between omega-6 and omega-3. Omega-6 is making more of pro-inflammatory eicosanoids. So we need to balance our omega-3. So if we balance them equally, our genotype is the best in the world to make longer chain PUFAs.
Yeah. Yeah, I got it, that answer, sir. One more thing correlating with that, which means you also mentioned about the COVID where inflammation is, can we say it like the people who get into cytokine storm or complication of COVID is more in a US population when you compare with Indians? Because Indian has a better conversion ratio, and we have, if we reduce omega-6 in our diet, can we not get that complication?
Yes, it's another good question, and it's a very hard question to answer and to convince a large number of audience.
Yes.
Because in US, what we are seeing now, the spike in cases, very much due to the Delta variant. We cannot say like, in US, the balance of omega-6 to omega-3 is 10:1. But in India, it increased, some data shows, Raheja et al., even he said 20:50.
Yeah.
So, we are eating a lot of omega-6 in our diet. If we can cut short omega-6 in our diet and take more omega-3, then our genotypes are better in conversion and making, less pro-inflammation. But with our excess eating of omega-6, now we are at a very high disadvantage when compared to other parts of the world also.
So only if we cut our omega-6, we are in the safer side in the genetical aspect as well as in the pro-inflammatory part.
Yes.
Yeah. So omega-6 rich food like refined oils and which is maybe largely found in omega-6 has to be cut down largely in our diet. That's what
that's what
take-home message.
Take-home message is, yes. Refined oils, as well as if we see some of the dietary oils which are in India, they are having very little amounts of omega-3 in them. They have large amounts of omega-6. The oils which we deep fry now in Indian cuisine.
Sir, for balancing omega-3 and 6, because it is an essential fatty acid, is it enough that, for balancing, we don't take omega-6 for a period of time, then it gets balanced because it will become deficient in our diet, hence the...
because we need a homeostasis, because both pro-inflammation and anti-inflammation is necessary for our body. And the DGLA is considered to be, again, anti-inflammatory, which is on the omega-6 side. If we decrease it too much, there you're having, again, an imbalance.
At least a particular period of time that we don't take it for omega-6 based diet more, that will help.
That can help in a short term, but not in a long term.
Okay. Sir, one more question that your another project on international HapMap project on Japanese, they have low FAD1 expression. You mentioned that. Yeah. So is it because of their food is rich with omega-3? Because they
actually, what we have seen, the lower expression in only 24% of their population, which is a minor allele, the DD genotype only, not in the II genotype.
And also, sir, any data available on Eskimos, because they take large amount of omega-3 fatty acid in their diet as a fish, their staple food?
Yes. Eskimos, they have a very high, one of the polymorphisms what we have shown in our 2016 paper, we measured one fatty acid, one SNP, which is following our Indel, but we haven't done much work on that. It shows like, they have, the DD genotype can be very, very high in Eskimos. And we have seen in Eskimos, the heart disease rate is very low in them.
So that maybe.
So they are there.
Yeah, they are in the safe side.
Yeah, they are protected here.
Yeah. Okay. Yeah. Thank you, sir. If there is any question, I will write to you, and we can get some answer from you. So from the audience, largely part we are missing it, but I will take the questions from them, and in that I will. Ma'am.
Yeah. We will post, in case if we get any questions, we will pass it on to Dr. Kumar and then post it in the YouTube later. Yeah. So thank you so much. Thanks a lot for this very informative session, Dr. Kumar.
Yeah.
And we look forward to have a few more sessions like this from you. We will keep in touch with you. Thank
Definitely.
you, Dr. Arun.
Thank you, ma'am. And I will take this opportunity to thank Dr. Kumar Kottapalli sir for giving his time and presenting the genetic aspects of omega-3. We were knowing that omega-3 and omega-6 are very essential fatty acid in our diet, and now we know how important our genetic plays. Yeah, and we, especially the Indian, we have better advantage. That's what I could understand from his talk. But we are not utilizing it because of the bad dietary habit, inclusive of too much of omega-6 in our diet. Maybe our guidelines and things and educating patients regarding omega-6 that we have to take it a little less, then you are in a better safe side in many aspects related to inflammatory disease. So very beautiful presentation, and I thank from our university and from our side and all the viewers. Very big thanks to Dr. Kumar Kottapalli sir. Thank you very much, and I also take this opportunity to thank Staff Development Director Dr. Deshmukh ma'am as well, and all other who joined for us all the 3 webinar series, and we will post this soon in the YouTube, and if you have any questions, and we can take it from there. Thank you very much. We'll see you soon with some other program.
Thank you.